I think we're getting started. So good morning, everyone. Thank you so much for joining us today. My name's Katie Boateng, and I'll be doing more introductions in just a couple of minutes. But today, the Smell and Taste Association of North America, in partnership with the Michael J. Fox Foundation, is bringing you this webinar called Could Your Nose Know? Smell Testing and Parkinson's Awareness. And we're so excited that you're here with us today. So our agenda for today's webinar is listed here. We'll start with introductions so you can get a sense of who's here with you in the room today, the virtual room. We'll talk a little bit about STANA. We'll talk about the Michael J. Fox Foundation and their Parkinson's Progression Markers Initiative, or PPMI. We will discuss clinical insights into smell loss and brain health. And then we'll end with a patient panel talking about experiences with Parkinson's disease and smell loss and personal experience with participating in the PPMI study. And at the very end, we'll have time for questions and answers. So some housekeeping for you so you're aware of how it will work today. We are in a webinar format. So the webinar will be recorded and it will be available to everyone who registered after the fact. So you can watch it again if you at the question and answers portion of the webinar. Please do note that if you use the raise hand feature, that this will make your name visible on the screen. So we just want you to be aware of that. Otherwise, you can use the Q&A box. There's no chat feature today, but you will be able to use the Q&A box at the end of the meeting to ask your questions. Finally, if you need anything with Zoom issues or you're not hearing us or anything like that, Courtney Pico from the Michael J. Fox Foundation will be available throughout the meeting if you have any questions. So I believe that you can ask her through the Q&A box. Is that right, Courtney? Yes, thumbs up. Okay. So we'll start with introductions and I'll just briefly let you know who's here today. So as I said at the beginning, my name is Katie Boateng. I'm the president of STANA and a founding board member. I also have acquired anosmia due to an upper respiratory infection for over 15 years now. We also have with us today Caitlin Kelleher from the Michael J. Fox Foundation. She's the Associate Director of Research Partnerships. We have Dr. Zara Patel. She's the director of the endoscopic school-based surgery, endoscopic sinus and school-based surgeon, and a professor at Stanford University School of Medicine, and a well-known specialist in smell disorders. And then finally, we have Don Upson with us today as well. He is a PPMI participant, a patient advocate, and he also has acquired anosmia. So we'll be diving a little bit more into his story today. So briefly, I'd like to tell you a little bit about STANA. Pardon my voice. So we're a nonprofit organization that founded in 2021, and our vision is a world where taste and smell are valued as essential to health, safety, and wellness. So we really advocate for people to have a better, improved quality of life who are dealing with smell and taste dysfunction. Our mission is listed there. I won't read it all out to you, but you can review that at your leisure. But really, we just want to improve the lives of people who are dealing with these issues like smell and taste loss or dysfunction because you're probably aware that a lot of people don't focus very much on these types of disorders. So we are partnering today with the Michael J. Fox Foundation to bring you this webinar because we want to discuss the PPMI study. There's a link between smell loss and brain disease that's well known, and we'll be discussing that today. So if you would like to, throughout the webinar, you can visit www.mysmelltest.org/STANA to learn more information, and we'll talk about that at the end as well. So now I'm going to pass it over to Caitlin Kelleher with the Michael J. Fox Foundation, and she's going to let you know about MJFF and the PPMI study.
Great. Thanks, Katie. Hi all, and I think Courtney is going to pull up my slides for me because I am terrible at sharing via Zoom. But by way of introduction, Katie already introduced me a little bit. My specific role here at the Michael J. Fox Foundation is on the patient engagement team. And our job is to think about people living with Parkinson's, take any patient insights, and then bring those into research development, study development, and then more specifically to help bring the right people into different studies, including the one that Katie mentioned, the Parkinson's Progression Markers Initiative. So just wait for these to load. Perfect. Great. Okie doke. So like I said, here from the Michael J. Fox Foundation, going to be talking about a little bit of our history and then how that relates to the study that we are hoping to get people interested in. So the Michael J. Fox Foundation was founded in 2000 by actor Michael J. Fox. And I think that what's important here is many people know who he is, but importantly, he is a patient. And he founded the foundation uh, for other patients. He and some of his colleagues and people in his network had this idea, which was true, that science was moving too slow in the Parkinson's world, and drugs were taking too long to be, you know, developed and approved and finally get into patient and family hands, which is an issue when you have a progressive neurodegenerative disorder. Our goal, you'll see at the top here, is that we're here until Parkinson's isn't. Our primary aim as a foundation is to go out of business. We want to end Parkinson's and no longer need to exist. To that end, we have funded more than $2.5 billion in research programs to date. So over the past 25 years, 88 cents of every dollar goes directly to research. We have hundreds of active grants, and we are reviewing thousands of proposals annually. Something else that I want to mention is you'll see here that it says we fund projects from academics, biotechs, and pharma. All of our funding is institution agnostic. We think that the best way to find better treatments for Parkinson's and eventually end the disease, cure the illness, is to get everybody in the room, as many people in the room as possible, which I'll talk a little bit more about later as we're talking about drug development and how we can really speed treatments and cures. The other thing I want to mention here is that you'll see over 40% of our projects are led by researchers outside of the US. When we say that we want to end Parkinson's, we mean for everyone, not just people living in one place or people of one background or creed. Next slide, please. Here we're chatting a little bit about the goal of drug development. So I said, part of our role, my team, is to bring the right people into the right studies. And this is really important when we're looking at drug development, where we want to match the right person. So somebody with the right symptoms, the right background, the right brain chemistry with the right therapy or new drug at the right time. Studies are very specific about when people can join them, the eligibility criteria, so what might make you able to join versus not join. And we really are trying to make it so that we are finding more of the right people for more of the right therapies at the right time. And this all is related to what MJFF does and where we see ourselves in the drug development pipeline, which I will now talk about. Next slide, please. This slide here is showing you essentially how a drug gets developed. So when it goes from somebody thinking about what drug could be developed through figuring out how that drug is going to work in the human body, testing it in the human body, testing it in many, many people with a disease, and then all the way through to regulatory approval by the FDA in the US or outside of the US, whatever governing body there is. On here, you'll see two blue sections. So the first is discovery. The US government and academic institutions typically are funding discovery. So that's, who has an idea for a drug, the very early stages of that. Michael J. Fox Foundation is in the middle in this orange section. So from figuring out what exactly we want to be targeting, translating that into the actual drug, and then testing it in humans for the first time in phase 1 and phase 2 trials. And from there, we then look to our friends in biotech, pharma, other funding bodies to try to bring things into even more people. So phase 3 trials, very large clinical trials, and then eventually through to regulatory approval. We see ourselves here in the middle because this is oftentimes referred to as the valley of death in drug development. There are things that are great ideas, academics are working on it, and then there is no funding in that middle period. So it will sit on a shelf for many, many years until somebody decides, oh, this is a good idea. We have the money to fund it. Before the foundation existed, it took 24 years for Parkinson's drugs to go from development to market. And frankly, that is a really long time, depending on, the human lifespan. It is far too long for people who are actively living with a disease or may develop a disease. Since we got involved, this has decreased by 50%. So now it takes an average of 12 years for a drug to get to market in Parkinson's disease. This is still too long, and so we're trying to find other ways to de-risk the field. The way that we do this is, like I said, funding, and then also, like I said, bringing the right people into the room. So trying to take a really bird's eye view of everything and saying pharmaceutical companies should be involved, academics should be involved, funders should be involved, and trying to figure out who are the right people to bring into that room so that everyone knows what the next steps are. That's sort of the largest thing that we do to de-risk. And then also we try to provide tools, we try to provide opportunities for discussion and other things that will make it easier for folks to move processes forward. Next slide, please. So one way that we do that is through our own research, which is what we're here to talk about a little bit more. We have a very large study, as Katie mentioned, the Parkinson's Progression Markers Initiative. The study has been around for 15 years. We're in our 15th year now, started in 2010, 10 years after the foundation was created. And the main purpose of this study is to try to find markers of disease for use in clinical trials. So if you know that somebody has a particular marker of Parkinson's, then you can target that marker in a clinical trial and bring the right people in so that you can see whether or not a drug is effective. Up until a few years ago, there were no solid biomarkers for Parkinson's, which I'll talk a little bit about in a moment. And the way that we are trying to find these markers is by collecting as much data and biosamples from people with Parkinson's, at risk for Parkinson's, and control volunteers, so people without disease or family history of disease, over at least 5 years. The study has been around for 15 years. There are some people who have been in the study since 2010, and people have continued and joined each year after that. As of right now, our goal is to have at least 5,000 people enroll. There are 50 sites in 12 countries around the world. The US is just one of those places. We also are enrolling people online for data collection. And this is something that is very important, although we're not going to be getting necessarily some of those biological samples. Really great for us to know what's happening when people are not able to come into a clinic, to have some other information about how their disease or their risk of disease might be moving forward. All of the information from the study is de-identified and is shared with the research community. So many of you on this webinar today could request access to this, could look at that data and see what might be interesting to you. And I do see a question in the chat, which I think we will get to in a little bit. So we will come back to that. I just want you to know that we do see it. This is just a timeline of the PPMI enrollment. So how many people have, or how we have thought about the people that we need to bring into the study. So you'll see 2010, we had our first participant enrolled. At that time, we were really focusing on people with Parkinson's and control volunteers. In 2013, so 3 years in, the study realized that we needed to also be looking at people who were at risk for the disease, not just people with the disease, because if we're trying to understand why Parkinson starts and changes over time, we actually need to be looking at folks before they develop the first symptoms. This continued for 8 years until 2018. We closed enrollment at that period to look at the data, think about what are the next steps. COVID-19 happened. In 2020, we did reopen enrollment for both people with Parkinson's and at-risk cohorts for even more sites. So prior to that, the 50 sites were not all open. There were fewer sites. The online portion that I mentioned launched in 2021. And then this year, 2025, we started enrolling even more people at risk and more people with Parkinson's to try to understand what makes people at higher risk for Parkinson's, what Parkinson's might develop like in different people with different risk factors, and then eventually develop drugs to treat all of those things. Next slide, please. Okay, so I was talking about how we're looking for biomarkers in this study and how up until a few years ago, we did not have a solid biomarker. And by solid, I mean like a good biomarker, not a physically solid biomarker. And through PPMI data and all of the people who contributed to the study, so the 3,000 or so people who contributed up until that point, we discovered a marker for Parkinson's in cerebral spinal fluid. And this little diagram is essentially just showing how you detect this biomarker in cerebral spinal fluid. But the gist is that in a very large portion of people with Parkinson's or with certain risk factors for Parkinson's, there's a protein, alpha-synuclein, that folds abnormally in your CSF and then potentially could be affecting your dopamine production and all the things that we associate with causing Parkinson's. So we found this biomarker, or the scientists found this biomarker, and now it is being used to try to bring more people both into PPMI, but also as a marker in clinical trials. So this is a really good example of how we are trying to de-risk the field by providing better ways to find out who should be in studies at what time, and then figure out if drugs are working. Next slide, please. So that biomarker is in your cerebrospinal fluid. But we're all here today to talk about smell loss and to talk about the connection between smell loss and Parkinson's and other neurodegenerative disease and what that means. So when looking at people who had this misfolded protein, this misfolded alpha-synuclein protein, we also were looking at smell tests. So over 30,000 people had smell tests, and a quarter of those had significant smell loss. And I know a lot of people on the call today may have significant smell loss. If we then correlate that data with people's imaging scans in your brain, looking at dopamine loss, the vast majority also had dopamine loss. And then one step further down the chain, if you had dopamine loss, you were more likely to have this abnormally folded protein. So what we started thinking about a few years ago was, can we use smell loss instead of CSF as a first cerebrospinal fluid as a first step to bring people into the study and then further look at their information, determine their risk for Parkinson's, see if there's other things there that we can be looking at. So we started doing that. We are screening a lot of people using a smell test, which we'll definitely get to more. Don is going to talk a little bit about this. I know his experience in PPMI and smell testing. And so we're using smell testing now to identify people who might have other biomarkers for Parkinson's. I do want to caveat this by saying just because you have smell loss does not mean you're going to get Parkinson's. I know Dr. Patel is going to talk about that, but I want to make that very clear right now. It's just something that is part of this mix of things that seems like it could potentially put you at a higher risk for developing this. Oh, and I do see here a question that I'll answer now, which is, has the same biomarker been found in skin samples? Yes, thank you so much for asking. The same type of protein has been found in skin samples. We're also looking at it in other types of samples. So nasal cells, retinal cells, other things along those lines. Blood is something that would be great to use as a test. As of right now, the tests that exist that look at the skin samples are not as accurate, essentially, as the one from the cerebral spinal fluid. So there are tests out there. We are looking at them in PPMI. So trying to see if there are ways to make them better, to make them more accurate at telling us who does or doesn't have this abnormal protein. But right now, the best way to do it is from CSF, which is frustrating because that's a lumbar puncture, which can be sort of scary, but also it's just the gold standard that we have right now. And I have my last slide here, which is just as we're trying to learn more about smell loss, trying to learn more about the connection between smell loss and other biomarkers for Parkinson's, smell loss and brain health, we are looking to enroll as many people as possible to take a smell test. So if you are 40 years old and you don't have Parkinson's, you can take a smell test. If you are 30 years old and you have Parkinson's and you've had it for 7 or less years, you can also take a smell test. If you don't fit into either of those categories, I would welcome you to think about the online portion of PPMI. We are constantly using that as ways to find people for other studies, and then also as PPMI's eligibility criteria changes, bringing more people into the study. So I'll stop there. Thank you very much. And I'm going to turn it over to Dr. Patel, who hopefully can provide more insight into smell loss, brain health, and why we're all here.
All right. Thank you so much, Caitlin. That was so wonderful. I'm going to go ahead and share my screen now, everybody. Um, Give me a moment here to bring this up for you. All right. Can you all see that okay? Okay, great. So I think that that was just a really excellent overview of the opportunities for patients and relatives on this call. And I'm going to take a little bit of time and really talk about both smell loss specifically and its link to Parkinson's and both what we currently know and what we're hoping to find out soon in the future. These are my disclosures. None of them are relevant except for Ulfera Therapeutics, which I will discuss briefly at the end because I think it would be really interesting for this particular audience to hear what we're doing in that company that I've co-founded. But just to make sure that I'm totally transparent and that that's my disclosure. So, a lot of people in the lay public really just started thinking about smell loss when the pandemic hit. But many of you on the call have been with it for many years. And certainly I have been thinking about it and studying smell loss for the last 15 to 16 years of my life since I finished training. Because, in addition to seeing patients specifically for smell loss, the way that I eventually became interested was because I started by just doing sinus and skull-based surgery, being an expert in the nose and the pathologies that happen in the nose and things like chronic inflammation in the nose and tumors growing in the nose, like this one I'm showing you here, certainly impact smell. And I could help all of those people that came to me with those issues. But there was this slow trickle of other patients that were coming to see me with smell loss, and we didn't really know why. And so that is really the category that Parkinson's disease patients eventually fall into, and I'll show you that. When I, talk about smell loss, a lot of people that don't experience it themselves don't have a great understanding of why it's such a big deal. And again, those of you that have it already know this. It's of course one of our evolutionary defense mechanisms that help protect us. We can smell smoke and escape fire. We can smell if food has gone bad and not eat it. But also the social impact of not being able to smell. And as many of you know, about 80 to 90% of our ability to taste our food is dependent on our ability to smell. And so it's really ability to smell and taste really impacts your ability to gather and socially interact with other people across all societies and cultures, any geographic region around this world. The way that human beings generally meet, whether it's with strangers or friends or family, it's almost always over food. We share food and drink and we meet and engage. And if you're not able to enjoy that or participate, or you feel somehow separated from that experience, you can imagine how that socially isolates you even further, and people start withdrawing, and that can lead to depression. And that's really what we see when we ask patients with smell loss from whatever the etiology is, whether it's a post-viral infection like Katie experienced, or whether it's Parkinson's, or whether it's any other reason, there's a whole host of different ways in which this can impact you and your sort of emotional and mental well-being. And, what I think is really important to note is that mortality in anosmics meaning just people who do not have the ability to smell, is almost 4 times more likely than in normal smelling individuals. And some of that is likely because of this link to neurodegenerative disease. I'll just talk briefly more. I could talk for a whole hour about all of the social implications that smell has for us, but this is a funny picture from a study back in the 60s. But there have been lots of really intricate studies done since then showing that, the way that we make first impressions of other people, but also the way that we choose romantic partners, sexual partners, lifelong partners really is completely dependent on smell. Now, this is going on a subconscious level, not something that you're really aware of. You probably think that you chose your partner because of the way they looked, or how compatible you are, or how successful they are, things like that. But really, we know that there is a part of our immune system called the MHC complex, the HLA complex. And women subconsciously have the ability to sense that, and they preferentially choose a person with an HLA complex that is as different from theirs as possible and choose those people as their mate. And so, that really is responsible for the diversity of the human species, and it really all goes back to smell. Another really interesting part of the way that we engage with each other is maternal infant bonding. And there's a really interesting study that shows, uh, mothers being presented with nappies and being able to identify and have a preference for their baby's nappy compared to all the other nappies they're presented with, unless that mother has postpartum depression, in which case she loses the ability to know which one is hers or prefer any to any others. And so I bring these little bits up just to sort of talk about how impactful, even on a subconscious level, smell and taste are for our quality of life and well-being. And I probably don't have to really emphasize that anymore to you on this call. Something else that's really interesting about smell is how many genes we have as human beings that actually code for olfactory receptor neurons. We have 6 to 10 million neurons on either side of our nose up at the top that help us smell and taste. And you can see that of all of those neurons, we really just have about 400 different types. You can see on this chart, as we're kind of on the bottom of this animal ladder, because maybe we depend a lot less on our smell to get through our day to day. But what's really interesting to me is what's at the very top. And you might think the African elephant has the most genes because it simply has the biggest nose. But what we also know about elephants, I'm just going to pause and let everyone think, what do you know about elephants? We also know about elephants that they have excellent memory. They have some of the best memory out of any other animal that we've studied in the animal kingdom. And I think there is a significant link there. When National Geographic originally sent out this survey to their subscription readership in the 80s and just tried to figure out like who is affected by smell loss, you can see that in the population over 53 years old, up to a quarter of patients had some form of smell loss. And that equaled about 10 million people in the US with total loss of smell even before the pandemic. The pandemic added about 15 million more, and that's just in the US. There's obviously millions more around the world. And we used to not really know why a lot of these patients had smell loss, but now we actually may have a clue. This was a really interesting study published in 2018 by my friend, Professor Hummel in Germany. And you can see that in this cohort of people that present to a smell clinic with smell loss, and we don't really have a good answer. There's no known virus or cold or sinus infection or trauma or known neurodegenerative disease. When they present with smell loss, we see that of that group that has combined smell and taste disorders, if you follow that group over time, a third of those patients eventually present with Parkinson's disease. And so, the time also that took for patients to present, the mean average time between when they presented their smell loss and when they had their diagnosis of Parkinson's was on average 10 years. So now you can really start understanding that we can use smell loss as the earliest sign before any of those other signs took place. And that's why we're so, why we're having this webinar and why we're so excited about the relationship between smell and neurodegenerative disease. I'm just going to briefly take a moment because I don't have time today in this brief little webinar to talk about all the different treatment options out there for smell loss. And I hope that all of you know that there are potential treatments. I know a lot of people go visit their primary care doctor or even a general ENT, and they're told, well, there's really not much that we can do, but there are potential treatments. So I'm just putting up my website so that you can maybe screenshot this or just make a note. And you can see that second tab is treatment options and evidence. And I've listed all of these things that we've studied and links to the study. So if you have more time and you have interest, please go see there. And I know not everyone has the ability to see a smell specialist that's close to them, but you can bring this to your doctors because there's so much that they can just easily do with you in clinic. So briefly to just touch on how in general does smell taste happen and how does that loss occur? You can see that we have odors that are in the environment all around us. Those odors come into our nose. They hit these 6 to 10 million neurons that I told you about up at the top of the nose. That sends a signal to the olfactory bulb that's just above the nasal cavity, above this plate of bone called the skull base, the cribriform plate. And from the olfactory bulb, that signal is sent back to the olfactory cortex, what we call the entorhinal cortex, and all of these very important connected regions, which if you know much about the pathology of Parkinson's, is very close and integrally related to the regions that are affected in Parkinson's disease. And, when we think about how does Parkinson's disease happen and how is this related to olfaction, many of you may be really, sort of familiar with what we call the Braak staging. And this is sort of the progression of the parts of the brain that are affected. And again, there's not enough time to talk about all the different theories out there as to how Parkinson's happens and why it happens and all the different mechanisms. There are so many. But one of the leading sort of theories is that there is an immune dysfunction and an inflammation or autoimmune inflammaging that happens in the brain is the sort of like key term that we talk about. And not being able to control or clear this inflammatory response may be interacting with different regions of the brain. And I want you to just focus on the brain images on this slide because I want you to see that the earliest stages, and this is because of postmortem analysis that we know this in patients who have had the disease, the earliest areas where we see this is in the olfactory cortex and autonomic system. I'll just briefly show you this picture of my grandma. My grandmother was a wonderful, beautiful, kind, gentle soul. She did not have Parkinson's disease. She developed Alzheimer's disease. And I bring this up because Parkinson's disease is just one of many neurodegenerative disease states that we know can be related to smell loss. And actually, the Braak staging system is true for Alzheimer's disease also. So Parkinson's disease, Alzheimer's disease, Lewy body dementia, there are so many different ways in which we can see this olfactory cortex being the earliest sign. So why can't we utilize that? Why can't we do something about it? Well, certainly you've just heard that we're trying to. We're sending out all these smell tests and there's a lot of good data now that shows if you pair that smell test with these already known tests for both Parkinson's and Alzheimer's, you can improve the prediction of development from mild cognitive impairment to these disease states within a short period or within a sort of 4 to 3 year period of time, depending on the disease state. But I can give you even one step further back than that. In the largest longitudinal study on aging in the US, this NIA Baltimore-based study, if you take healthy aging adults and you give them a smell test, for each point lower on that smell test that they score, there's a 22% higher chance of developing mild cognitive impairment. So again, this really shows how early this smell loss can be compared to everything else that happens. This is just to show you guys what Caitlin already kind of touched on, kind of giving you a little bit more of the granular details. I want you to look at these two little areas. I know it's a lot of detail on this slide, but this is from this, Parkinson's Progression Markers Initiative. This is one of the studies that has been published in 2023, I believe. And you can see that, you know, when you look at all Parkinson's cases, right above that red arrow, you can see about 87, 88% of patients are having this important biomarker. When you pair that with smell loss, which is what hyposmic means, you can see that really bumps it sky high, 97%. When you go further down and you look at these different genetic markers that we know can predispose people to Parkinson's, you can see that the GBA mutation has a pretty high percentage of those patients that have this biomarker across the board. But that LRRK2 mutation is lower. It's 67.5% in all patients. But if you look even further down in that white box down low, again, if you pair smelling ability with the LRRK2 mutation, you can see that again bumps it back up to 89, 90%, predictability of Parkinson's. And so this biomarker paired with smell loss is really important. That's why we're asking and sending out all these smell tests to everybody. So again, briefly, just to touch on the therapies that I don't have time to talk about, but I hope you guys all know this is what can work to bring smell and taste back. These are therapies that I'm often asked about and do not offer. You may have heard about some of these things in the media. They have either been studied and shown to not help, or they've been studied and actually can cause worsening of smell. So please don't try those. And then I'm just going to briefly talk in my last few minutes about, the future. I'm working a lot on electrical stimulation. And why would we think about electrical stimulation? Well, we know that peripheral nerve electrical stimulation really leads to acceleration of axon outgrowth. That means growth of neurons across injury sites and reinnervation of end organs. All of you are probably very familiar with deep brain stimulation. It's used very commonly in Parkinson's disease to modulate neural circuits that are not functioning correctly. Modulating neural circuits, ablating incorrect signaling, and allowing correct signaling to take place again is one very common use of electrical stimulation. And specificity of target is actually something else that we've been able to see can happen when you stimulate nerves that have multiple different functions. If they're both cutaneous and sensory and they're motor, you can reinnervate and regenerate in the exact right way whereas without the electrical stimulation, that reinnervation is much more random and chaotic and maybe not correct. Who might benefit from that? Like all patients with smell loss, basically, including you guys. This is just an animal study that I had done showing that you can improve food finding times in rats, and you can see the difference in the olfactory epithelium that happens when you regenerate. And for a precision medicine way to stimulate, the key here is that first we need to record. And as you all know, and what we've been talking about, and the type of smell test that we're currently sending out is this sort of scratch and sniff smell test. Now, I want to take a moment and say this is great for what we have right now. However, in the last sort of, compendium of all olfactory clinical knowledge that we put together in 2022, this is the 20-page table of all the smell testing methods that are out there in the world. And you know, when there's this many different ways of testing smell, that means there's not one perfect way of testing smell. And the reason is that all the methods we currently have that we can use clinically are subjective. They're time intensive. When I give someone the smell test, I'm hoping that I know whether they are actually identifying the smell. But what we might be testing for is whether they're familiar with that smell. If they grew up in that area that smell is present, whether they are literate to fill it out on their own, whether they have the same language, whether they have the cognitive ability to understand the task. And so that's why I'm, developing this device right now that can potentially endoscopically, meaning through the nose, directly record from the olfactory epithelium all of those nerves and give us a very precise, in each individual, precision-oriented score of how well people can smell and specifically what is missing in their smell ability. I've done a lot of self-experimentation and miniaturization and help from people here at Stanford. We just met for this time, this like group of just about 20 of us around the world that are working on electrical stimulation of the olfactory system. You can imagine that this stimulation is not going to just have effect on the olfactory epithelium, but hopefully even further into the brain. And then briefly, just to talk about that company I was telling you about, Olfera, utilizing the olfactory system to actually deliver drugs through the olfactory nerves directly to the brain. As Caitlin mentioned, there is so much problems with translating what has proven to do really well in the lab into patients. And part of that is because we have to, get drugs through the gut or through the bloodstream into the brain. And there's lots of barriers. If you could directly put a drug from the nose through the olfactory nerve into the brain, that would be amazing. And so that's something we're working on for all of you. I know that was a lot of information. I'm happy to take questions. And if we don't have time for them all, this is my Instagram and you can message me there.
Thank you so much for sharing, Dr. Patel. Now I'm going to share my screen again. One moment here. And we're going to move on to the portion where we're going to speak with Don Upson. He's going to discuss with us his personal experience with smell loss, being a PPMI participant, and having acquired anosmia. So, Don, to get started, before we speak specifically about the PPMI study, I'd like to know a little bit more about your experiences with smell loss. So, can you share a little bit about what you have experienced?
Sure. My smell loss became quite obvious in my mid 40s. This is some time ago. And here we are 35 years later, and I can smell very, very little. It's kind of a joke almost because, of course, smell and taste are related, and I'm the guy that's always saying, "Pass the hot sauce," because be able to, have some sensory um, input for this. So all of my tastes, all of my smells are virtually zero. My tastes are severely blunted. And I had actually kind of attributed that to my career in chemistry, thinking, oh, maybe I got exposed to some toxic materials that caused it. And it may have played a role, but that was just an idea that I had. Anyway, until 2015, and the significance of 2015 will become apparent here very soon, I didn't know that smell and taste loss and Parkinson's were connected, or even that the Fox Foundation had formed PPMI. So anyway, in 2015, our then 37-year-old son was diagnosed with early onset Parkinson's disease. That hit me like a ton of bricks, like I think it would any parent. I pledged to do all I could to help him and others by being part of the solution. And so I looked up the Fox Foundation, was very impressed, and I contacted them and decided I wanted to try to join PPMI. Well, what I learned was that the first participation step for PPMI was the smell test. And I took it, and I think that I probably scored something like 25% on it because there were four possible answers for each, and I could not smell a single one of them. So they told you to guess. So I probably got 25% just because. Anyway, the smell test categorize, categorizes the undiagnosed people as either normal or as prodromal. Now, what does prodromal mean? That's a sort of a $700 word, but it means having a risk factor or a symptom, but not yet diagnosed. And as was properly pointed out by our earlier speakers, having a risk factor does not necessarily mean that you have the disease or even that you will get it. It just does mean, though, that you're a little bit more likely to get it. So I think it was also pointed out that there's about 5,000 people now participating in PPMI. And my understanding is that even more are being sought. And this includes normal and prodromal patients. Okay, so I'd like to move on, if I can, into what is it like to be part of PPMI? Well, as a Parkinson's prodromal, I got actively involved fully in PPMI very, very quickly. For the last, well, this is year 5, I have been taking these all expense paid trips to PPMI clinical centers. For the first several years, I went to New Haven, Connecticut, where I did that in the shadow of Yale. And then this year, actually just last week, I did the same thing at the Mayo Clinic in Phoenix, Arizona. I live in Arizona, so that was quite convenient. Um, anyway, those are 2 very intensive days of diagnostic tests. And I've kind of lost all of my video on this, so I can't see what you're seeing. Uh, but if we could make sure that
Don, I
the
have the in-clinic tests performed on the screen for you.
Okay, terrific, terrific. So what you'll see there is that these tests fall into 3 categories. Uh, the first 2 are imaging for brain MRI and a brain DAT scan. Now, DAT is you're probably very familiar with MRI. You may or may not be familiar with DAT scan, which measures dopamine transporters in the brain. And so there's the imaging, then there's the physical sample collection, and that includes blood draw, urine collection, uh, cerebrospinal fluid collection, with the lumbar puncture, and a skin biopsy. And all of these specimens go to a library of samples that are available for research purposes, as I think was also already pointed out. Then the third category of testing are mental and physical capability testing. And you can see the list goes on and on. And so it's a very intensive day. There's almost no downtime. You're just going, going, going. And it's a little bit exhausting, but it's also uh, very satisfying to think that, okay, maybe my son will benefit from all of this. So now that's the clinical visit side. There are a whole host of things that we do in PPMI on top of these clinical visits. And those are also on one of the slides. There are quarterly emailed surveys, Zoom meetings with physicians. I have actually, this is my second time in a webinar, and I recently helped to make a TV ad that will encourage people to take the smell test that I believe will be aired sometime this fall. I'm a monthly donor to the Michael J. Fox Foundation. I send letters to Congress to plea for research support for Parkinson's disease. And lastly, very lastly, I have agreed to donate my brain post-mortem to be examined. And so that's kind of what I do as part of PPMI. You can do some, or whatever fraction of this you are willing to do and feel inspired to do. So the big question then comes is, why does all this matter? What is the significance? Well, you've actually already heard a lot of that, because from these tests, especially with uh, the spinal fluid and the skin tests, the alpha-synuclein marker was identified, I believe, last year. And that gives a 93% accurate, very early diagnosis of uh, Parkinson's disease, far faster than was previously possible. And as Dr. Patel emphasized, early detection really matters. So ultimately, now I get into a little bit my science background, I guess. Ultimately, a target molecule will be found that is both responsible for the disease and what we call druggable. Okay, that's a sort of a term that I like to use, means that research teams can take the target molecule and find drug candidates, which are at the beginning are called hits, that will partially arrest the disease. Uh, these results also can be used to better understand the initiation and progression of the disease. So the candidates that I just called hits will focus the research and clinical trials on developing more and more effective and selective drug candidates. Typically in pharmaceutical parlance, you might have a hit that would have uh, an effectiveness at a particular dose. And what you want to do is you want to keep lowering that dose as far as you can, but maintaining the effect. And what that does is that cuts down side effects tremendously because you don't have all this extra stuff floating around. So I would say that developing more and more effective and selective drug candidates is really the ultimate significance of PPMI. Arrest and hopefully cure Parkinson's disease. So I guess I would like to encourage everyone who has not yet taken the smell test or considered being part of some aspect of PPMI to go ahead and consider it. I would thank you, my son would thank you, and the thousands and thousands of people with Parkinson's disease would also thank you.
Thank you very much, Dawn, for sharing your story and your participation in the PPMI study. At this point, we have about 7 minutes left before the end of the webinar. So we have a couple of questions and answers that we can go through. So Caitlin, I wanted to ask you, there was a question in the chat. It says, is olfactory function currently used in drug development studies on Parkinson's disease patient enrichment? Would you mind answering that?
Yeah, great question. I'll do my best and then might lean on Dr. Patel if she has other things to include. So up until now, the study of sense of smell in Parkinson's has primarily been about progression. So just realizing that loss of sense of smell might be one of the first signs of Parkinson's. To my knowledge, not we, to my knowledge, industry groups, so pharmaceutical companies, other drug uh, purveyors have not used it for inclusion and exclusion in a trial, but they are often collecting the scratch and sniff smell test that Katie mentioned, the University of Pennsylvania smell test inventory or small inventory test, the UPSIT, as part of their data collection to try to understand the full range of somebody's disease. I think that it's possible in the future if we had an upset that was more specific to Parkinson's that they could use it. But right now, it really is just about general smell loss and not only Parkinson's. Dr. Patel, not sure if there's other, yeah.
Yeah, I completely agree. Uh, the short answer, like Caitlin said, is not really, not right now. However, there are more and more pharmaceutical companies and drug developers that are starting to understand this connection a bit more. And so, although a smell test may not be part of like an enrollment criteria for drug development, there are some basic science type of tests that we're doing. So for example, there's something called brain on chip that we can use where we see the olfactory neurons and system connected on a computer chip, and you can do a high throughput drug delivery through that and see what affects the olfactory system and what binds and doesn't bind. And so there are a lot of drug companies starting to think about the olfactory system and smellability um, as part of their drug development process. But the short answer is no, that's not part of enrollment criteria now for trials.
Thank you, Caitlin and Dr. Patel, for responding to that. So I would like to just remind everyone that you can visit www.mysmelltest.org/STANA to sign up and request to take the smell test. Just a reminder that smell loss can be linked to brain disease. It does not mean that you are going to go on to develop Parkinson's disease, and that anyone who's aged 40 or over, who's in the US and Canada, is eligible. Anyone who's age 30 and up with Parkinson's disease within the last 7 years is also eligible, but you can also check out the online resources that Caitlin shared. And with that, I would just like to say thank you so much to all of our panelists for participating today, and for everyone who's here watching. A reminder that we will be providing a recording so you can review this information at a later time. And thank you so much for attending today. I hope you have a great rest of your day. Thank you, everyone.